All. This is Dr. Mobeen Sayed from drbeen.com. Welcome to one more show. Hopefully, you're doing good. Um, the discussion that I wanted to do today was about the um, antibody dependent enhancement. However, I saw that there were results from Johnson and Johnson's phase 1 and 2A trial as well. And it made more sense to look at that so that we are getting ready to understand if we are okay with that vaccine or not, and what are the pros and cons of that vaccine. I had done a discussion about the phase 3 of the vaccine and the mechanism of action, or the build out of the vaccine itself a few days ago. I believe the title of the video was "Will you take this vaccine?" So if I can very quickly share my screen here. This is drbeen.com, and here is that video. This is titled, "Will you take this vaccine?" I have added to the title Johnson & Johnson vaccine, so it is easier to search. In this vaccine, I have talked about how this vaccine is built, what are the PERC6 fetal tissue clone cells, what is the adenovirus being used here, and so those techniques have been mentioned in this video. Today, what I wanted to do was I wanted to talk about the results of the phase 1 and 2. Phase 3 trial, which you are seeing here the video for, that trial is going to finish somewhere in another 2 or 3 months, and then early 2021, early this year, hopefully we'll have this vaccine available in production as well. Now, with that, this is the results in New England Journal of Medicine. These are the results for phase 1 and 2A trial. I'm going to go over those with you. This is the PERC6, the fetal tissue cells and their clones. This is that technology, AdVac technology by Janssen, which is a subsidiary of Johnson & Johnson. And then this is an important and interesting article where they have discussed various storage needs for various vaccines. So interesting to read. So with this, let's start with my way of presenting things. So Johnson & Johnson vaccine. So let's start from the top here. The vaccine's name is AD26. AD stands for adenovirus. 26 is probably their 26th vaccine based on this virus. CoV2, so the vaccine is for SARS-CoV-2, and S stands for spike protein. So this is an adenovirus-based vaccine for SARS-CoV-2 against its spike protein. Company, of course, Johnson & Johnson, and we discussed this in a few days ago, that Johnson & Johnson had acquired Janssen I think decades ago, and then Janssen had acquired Carousel or something, which had PERC6 cell line. So all of those companies together are the one that are producing this. So parent company is Johnson & Johnson. Vaccine type is adenovirus. And again, it is adenovirus, which is chimpanzee based. And many people comment on this particular point and say, I don't want to have a chimpanzee adenovirus. I would rather have a human adenovirus and not have this vaccine. Look, human adenovirus, we have become used to them. Our immune system already know them. So as soon as you put those adenovirus in us as a vaccine, our immune system is going to attack them immediately and kill them, and so or destroy them. So they're not going to be able to deliver the vaccine to our cells, and before that they will be eliminated. So human adenovirus are not a great vehicle to bring the vaccine in our cells. On the other hand, chimpanzee adenoviruses are better because our immune system does not know them, just like our immune system does not know SARS-CoV-2. These adenoviruses are modified. Their replication genes have been removed. So when they are injected in our body, they cannot increase in number. So if you inject 1 billion of them, they would only stay 1 billion of them. So that is the adenovirus-based vaccine. Then how is it manufactured? This vaccine is manufactured in the clones, copies of the PERC6 cells. PERC6 cells are fetal tissue cells. I believe these are the retina cells of a fetus who was aborted at 18 weeks and was healthy. And again, all those ethical, moral questions that come to your mind, we have been talking about it. I'm not going to indulge in those at this time. If you don't feel that this is something that you agree with, then this is not a vaccine for you. So just know that it is built in fetal tissues copies. Now, antigen production, what kind of antigen this virus or this vaccine will make? So the adenovirus that is injected in our body, that adenovirus inside has the messenger RNA or DNA actually for the spike protein of the SARS-CoV-2. So it is a spike protein that will be produced in our cells, which will then cause the immunogenicity, or which will cause immune response and reaction. Adjuvants, I think that there are adjuvants in there, but I could not find exact data to understand what they are. The reason I think that there are adjuvants is that I read a line somewhere while researching this, which said that they have the the adenovirus can act as an adjuvant so that the immune pathway goes T helper 1. So we'll look at that. But I'm not sure. So this is why I put a question mark. Availability. So early 2021 will this vaccine be available? And generally on the vaccines point, I am so disappointed at the way we have reached so far with the vaccine. So if I go here to the, let me close this one, Bloomberg vaccine dashboard. So if you see here, 15.6 million shots. We started on 14th of December. 14th of December to 14 January, about 31 days. And then we have another 5 days now. So 36 days. We were supposed to have distributed all 25 million initial doses within the first week of January. And then we would have started the other folks as well, essential workers too. So we are still behind. We are catching up. We are behind. I think we'll become fast as our gears, the initial logistics become mature and we learn how to do these things better. Then we'll become better. Although I think you may have heard that in California, they have paused a batch of 330,000 doses because there were too many allergic reactions with that batch. So there are those hiccups or dangers with the vaccine that are happening as well. Okay, so back here, availability early 2021. Storage, I think you would like the storage. This vaccine can be stored in refrigeration, normal refrigerator temperature, for 3 months. And if we put it at -4 degrees centigrade, then it can stay there for 2 years. So that is a very interesting point, because Pfizer is very, very temperature sensitive than Moderna, and this one is very less temperature sensitive. Dose. So this is interesting as well. The dose that they have tested in phase 1 and 2, which we are talking about today, after that testing, they have arrived at the dose of 5 multiplied by 10 raised to power 10 number of adenovirus particles in 1 milliliter of this vaccine. That makes it 50 billion particles per milliliter of the vaccine. That is a dose. And that is a dose that they have agreed on. So 1 milliliter of vaccine is injected. Response type. So we know that we have immune system response, right? Humoral response, which is T helper 2 based, or cytotoxic response, which is T helper 1 based. So this actually invokes both responses, humoral and cytotoxic, which makes sense. Almost every infection causes both arms of the immune system, adaptive immune system, to become active. It's the percentage of activity. In some cases, the T helper 2 pathway or the humoral pathway is dominant and T helper 1 pathway or cytotoxic pathway is not. And it can be the other way around as well. Excuse me. So T helper 1 and T helper 2 both are triggered by this vaccine. However, T helper 1 pathway, which is for cytotoxic T cells, that is dominant instead of the T helper 2 pathway, which is for humoral. And they actually tout this as a great thing because they think that because of this, the possibility of antibody dependent enhancement is not there. So that is only a theoretical thing which is only observed in some animals, never in humans. And because the T helper 1, which is cytotoxic pathway, that is more dominant compared to the humoral pathway, that is just not possible anyways. And I'll discuss that a little later. Animal studies, has this been studied in animals? So yes. And why do they say yes? Because the platform that they're using, this platform, AdVac and PERC6 platform, this platform has been used to make many other vaccines, and about 100,000 people have been vaccinated using vaccines generated in this way. And these vaccines have then been tested on Syrian hamsters and on non-human primates, that means chimpanzees and other monkey breeds. So it has been tested on animals. This is a very common objection on the vaccines as well, that hey, vaccines are not tested on animals. So this whole platform has been tested and the safety has been seen already. Now, what are the results of this phase? And I would actually share this. So this is the document. This is the PDF. This document is present here as well. But there is a summary of this over here. You can actually download the PDF. Here is the PDF. I have that PDF open here. And it is interesting to actually go over this PDF just for your own knowledge and curiosity's sake. So let's look at this PDF as well. And we'll look at the document in a second. First of all, the design of the study is that it is a multi-center study. So they tested it in multiple centers. Fine, that's okay. Placebo controlled. So there are some people who are in placebo arm and some who are in the vaccination arm. They had 2 groups of people that they recruited. So the first recruitment was from July 22nd, and they all got their first dose till November 7th. And the second recruitment was August 3rd and then onwards. And the first recruitment had 402 people in it, and then the second recruitment had 403 people in it. Remember, this is a phase 1 and 2A trial. These phases normally have smaller number of people because we are not yet sure that what is the right dose of the vaccine and if it is safe or not. So in these phases 1 and 2, we are usually trying to figure out what is the dose and the safety. Because of that, we do not give it to broader, bigger group of people. Now, there were 2 important cohorts or groups that were important. One was that they were 18 to 55 years of age group. That was cohort 1. So if you read this document, wherever they say cohort 1, that is the 18 to 55. And then 65 and over 65, they were cohort 3. So in this document, wherever they say cohort 3, that is the 65 plus. The endpoints, what were they trying to establish? The primary objective of this vaccination trial 1 and 2A was to look at the vaccine safety and reactogenicity, meaning will it create reaction in our body. A secondary endpoint was to look at humoral versus cellular immunity, meaning they were going to see if the antibodies are produced more versus cytotoxic T cell response becomes more. And I would explain that in a few seconds. So these were the endpoints. The doses that they used, remember phase 1 and 2 are early phases. In this, we are actually testing doses as well. So they had given a low dose, which was 5 multiplied by 10 raised to power 10 virus particle, adenovirus particle per milliliter. So that was 50 billion particles. This was low dose. And then the higher dose was 1 multiplied by 10 raised to power 11. So that is 100 billion instead of 50 billion. And that was the high dose. And then the third group was placebo. They had a fourth group as well, but anyways, cohort 1 and 3 are the ones that they talk about. So I've just put these two over here. This is the important part here. If you see at the bottom right of the screen, this is important. The importance is that after giving the first dose, after 29 days of that, the 90% of the individuals had developed the response to the adenovirus or a response to the spike protein, which means response, our body, 90% of the people had become ready to combat the SARS-CoV-2 after the 28 days of the first dose only. So that is a very interesting behavior. And this also tells us one more thing. Many people ask this question very often, that when is it that my immune system will become ready? So here, after giving the dose, it took people 29 days to develop the protection. So immune system is supposed to start working anywhere, adaptive arm, anywhere from 7 to 14 days. But here, if you're seeing 29 days. So that means when we get infected or when we have vaccine, it can actually take about a month for our body to become fully prepared for the virus. And that is why the SARS-CoV takes the advantage of that time. During this time, SARS-CoV-2 can actually cause lots of damage. Okay, so continuing. What were the results? So here is what they had done. The two cohort, the cohort 1, 18 to 55 years of age people, they had given them 2 doses. And the cohort 3, that was 65 years and over, they had given them 1 dose. Most common systemic side effect observed was fever. And people who received 2 doses, they had more fever than people who received 1 dose. Makes sense. Then headache, injection site pain. The most common localized reaction was pain on the injection site. Myalgias, that is the muscle pains, fatigue, tiredness, and then that's it. So these were the most common side effects. And how many people get those kind of side effects? About 60 to 70%. So that means if we all go and get this vaccine, and if we are 100 people, about 70 of us are going to have the pain at the site of injection. Many of us would develop fever as well. Many of us would have myalgias and fatigue as well. Some of us would have headache too. So these are the most common one, mostly 50% and above. Now, this is an interesting slide. I think you would like this one. So I see a comment here from Janet. She's saying, this is a lot to go through just to get a bio ID. Yes. Well, this is a lot that I had to research. So I spent a lot of time going through these things as well. And I highlight them as well to pull those points together. So I'll show you that document in a second. So let's see antibody titers. That means once the vaccine is given, the neutralizing antibodies. Remember, neutralizing antibodies are those antibodies that would bind to the spike protein of the virus, and they would not let it attach to the ACE2 receptor, and so they neutralize the virus. So remember, we have 3 kinds of antibodies: sterilizing antibodies. And I think this is one of the reasons that when we say sterilizing antibodies, people say, well, it's going to make us sexually sterile. Sterilizing antibodies simply mean that these are the kind of antibodies that when they attack the virus, we cannot transmit the virus anymore. So that these kind of antibodies are called sterilizing antibodies. And then if they simply attack the spike protein of the virus and make the virus unable to connect with ACE2, then we call it neutralizing antibodies. And if the antibodies are just binding with the virus, regardless of where they are binding it, they're not doing anything to it, then these are simply called binding antibodies. So here they were testing for neutralizing antibodies because that is the function of the vaccine. So here dose one after the 28 days of dose one, the antibody titer against the spike protein increased. So this is called, the GMT over here stands for geometric mean titer. So that is just a way to compare the titer to another standard titer. So it went up 224 to 354 in that range. Then at 57 days, almost 2 months, the titers went up to 288 to 488, and the titer became 100% immunoprotective. So after 2 months after the first dose, the person developed so many antibodies that they are protected. And these high titers stayed stable for 71 days. Well, you can say, why not more than 71 days? They could only test for these many days because then the trial finished and they had to give the data. This is interim data. This is not the final result yet. And they wanted to start the phase 3 as well. So the number of days they had, according to that, for at least for 71 days, they know that the immune reaction stayed stable. For those people who got second dose as well, I did not find the number of days after the second dose, but they say after the second dose, the titers actually, or the amount of antibodies increased 2.6 to 2.9 times. So almost 3 times increment in the antibodies which were developed after the first dose. So if you see here, 827 to 1266 geometric mean titer, a lot of antibodies. And that is a good thing because we can now capture the virus. So this is the antibody production. What is the takeaway for us here? After one dose, 28 days after, 90% of us will become protected. And then we will continue to become more and more protected for next 1 month. And if the second dose is given, then the protection goes up 2 to 3 times. So very strong reaction. Now, cellular response. This is what is interesting. You have seen this diagram many times. I would like you to see this diagram today from a different lens. The lens today is that, remember the antibody dependent enhancement is a concept that we talk a lot, and people ask about that a lot as well. And they feel that if they get the vaccine, they would get ADE and that would cause an issue. So I always say it, and I'll say it again, at least for the cool beans here, it is a theoretical possibility. We have not seen it in humans. It is observed in some models of mouse or other animals, and even those are specially prepared to check for this phenomena. So they are kind of artificially biased towards creating ADE. And the reason for that bias is to kind of observe and verify that, hey, this hypothesis of ADE occurring, will this occur if we give it the chance? But it is not actually observed in us with the SARS-CoV-2 or the vaccines. So that is a lens to keep an eye on. And now let's look at what will our body do when the vaccine arrives in our cells. So let's say vaccine is given. So we're starting with the left side of the diagram here. Vaccine is injected. Vaccine is lots of adenoviruses. How many? 50 billion adenoviruses in 1 milliliter. Those go into the tissue. And of course, the immune cells in the tissue, for example, what are these cells called? These guys, dendritic cells. You know that when I'm presenting, if I forget something, I forget something. Or if I switch it, then I switch it. I cannot fix it. My brain will not go back to the normal. So I get stuck in a particular mindset. So dendritic cells. There are dendritic cells here. There are macrophages here. There are fibroblasts here. There are other cells here. So what happens is once this vaccine is injected, our dendritic cells are going to pick this vaccine up and the immune system response will start. Meanwhile, macrophages will do it as well. And dendritic cells are kind of a type of a macrophage. Macrophages will do that too. And then in a few hours, these cells will travel to lymph nodes too. So the vaccine will be traveling from the deltoid muscle to the local lymph nodes, where there are B and T cells sitting who need to be, excuse me, who need to be activated for adaptive arm. So the innate arm cells are like policemen who would capture something and bring it to the jail or police station, and then the remaining process starts there. That is a similar kind of a thing here. In addition to the local lymph nodes, some of these cells are going to circulate in the blood, and they would bring it to the spleen as well. So the vaccine is found in spleen as well, and this would be probably true for all of them. Although Johnson & Johnson categorically mention it, that when we give the vaccine, we see the immune system cells take the vaccine to lymph node and to spleen, which is a good thing. These are lymphoid organs, these are immune system organs, and their involvement means vaccine will work and we would develop a response. So back here, vaccine is given, then the dendritic cell picks it up and presents it on the MHC2 part of their presentation. And here is a naive T cell. The naive T cell will come and connect with this one. And now if there is interleukin 4 present or absence of interleukin 12 is there, then what happens is that the naive T cell becomes T helper 2 cell. T helper 2 cell will in turn release interleukin 4 and 5 and 10 and others, and it would cause a naive B cell to become an active B cell or plasma cell, and that plasma cell will start making antibodies. This process in case of this vaccine takes up to 28 days. Now the same vaccine also causes this pathway to work as well. So what would happen is that the naive T cell is connected to a dendritic cell. The dendritic cells actually and macrophages release interleukin 12. So in the presence of interleukin 12, these naive T cell will become T helper 1 cells. And T helper 1 cells would release interleukin 2. I should write interleukin 2 over here. And these cells, interleukin 2 cells, will then cause, or the release of interleukin 2 will cause cytotoxic T cells to become active. And the cytotoxic T cells are also called CD8 plus cells. These CD8 plus cells or cytotoxic T cells are going to then connect with other cells that have the vaccine or the virus and kill them. Now, I had requested you to look at it in a different lens today, and that is this. When this vaccine is given, this pathway, the cytotoxic pathway, is more prominent and more dominantly triggered by the vaccine compared to humoral pathway. So T helper 1 activation is more, and as a result, cytotoxic T cell activation is more than T helper 2 cell activation, which creates plasma cell, which makes antibodies. Now, why is this interesting? This is interesting because when we have less plasma cells present, then the concept of theoretical antibody dependent enhancement is not very viable, because there aren't enough cells that are overwhelmingly producing antibodies, filling our body with the antibodies and making those antibodies a receptor for the virus. So that is an important thing to note, and this is what I have taken from the documentation. I'm not just making it up, I'll show it to you. So that is an interesting concept to keep in mind. So that is what I've written here. T helper 1 sides ratio was 70%, while T helper 2 sides ratio was 30%. So this is how dominant T helper 1 side was. So if you see here, on 14th day, 14th day. Remember, we talked about 28th day as well. So 28th day is when antibodies, that means the humoral response, became strong enough. Now we are looking at the 14th day, 2 weeks after the first dose. In the cohort 1, that is the 18 to 55 years of age people, 76 to 83% individuals had become cytotoxic T cell positive, meaning their cytotoxic T cells had become trained. I love this part. Do you know why I love this part? Not only this actually reduces the chance of ADE, I also love this part because we have seen from studies that those people, those patients who get the wild infection, that is the real SARS-CoV-2, and if they respond with T helper 1 pathway, the cytotoxic pathway, they actually handle the infection with lesser symptoms and with lesser chances of cytokine storms. So T helper 1 pathway, or the cytotoxic T cell pathway, actually seems to be a better pathway. And with this vaccine at the day 14, this pathway, the better pathway, actually becomes active. In how many people? 76 to 83% of the people within 14 days. Awesome news. And in the cohort number 3, where there are 65 years and plus, they are 60 to 67%. So lower percentage of people develop this within 14 days. They may develop it a little later, but within 14 days, 60 to 67. This is very interesting for me. This particular concept or observation by the team is very interesting for me. So this is the discussion. Let me now just very quickly go over some of the interesting points here. So this is the document for the phase 2. If you can see here, these are the pieces I've taken up. These they've discussed. This is the summary. Then if you go into detail, they have discussed the method, that how the people were chosen, how the doses were given. They have the ratio here. See, they had actually one. So if I go here, 1, 2, 3, 4, 5 groups. Out of them, cohort 1, 2, and 3 are the ones that we are looking at. Then if you see here, primary and secondary endpoints. So the primary endpoint was safety and reactogenicity. And the secondary endpoint, this part here, was humoral and cellular immunity, which we just looked at. Then they said, how did they do the lab analysis? Then over here, they're talking about the participants, and that is what I've already discussed. These tables are interesting if you are interested to see male versus female, age ratio, and so on. Similarly, ethnic groups and race groups, they have all of that data here. This is interesting. These are the side effects. So what are the most frequent side effects? For example, injection site pain, 64% of the people in the cohort 1, and then 78%, I believe, or 42% in cohort 2. So they have various side effects and their percentages here as well. Then they talk about the immunogenicity and seroconversion. Seroconversion means the presence of the antibodies, and the cytotoxic immunogenicity is cytotoxic T cell and B cell responses. So they have that data over here as well. So I generally like their presentation. Look at this one. However, all the participants who had a measurable T helper 1 or T helper 2 response had a T helper 1 to T helper ratio that was well above 1, which indicate a vaccine-induced T helper 1 skewed response. That means this side of the response was dominant. So if you see here, S specific, that is spike protein specific, CD8 T cell, that is the cytotoxic T cell response, as identified by the expression of interferon gamma or interleukin 2. So if you see here, what happens is cells 1, they release interleukin 2. So if you measure interleukin 2 and it has gone up, that means T helper 1 side is working. And secondly, when the cytotoxic T cells become active and the T helper 1 cells become active, they release interferon gamma, which activates the innate arm. So presence of interferon gamma is an indication as well of the cytotoxic arm that is working more. So that is what they're talking about here. expression of the interferon gamma or interleukin-2 cytokines on S peptide stimulation. So what they did was they took the cells and they stimulated them with the spike protein, and the response appeared on day 15. And so we talked about it. I just wanted you to kind of see it as well. This is a very interesting paragraph. They're talking about spike protein specific T cell responses, especially the T cytotoxic responses. So that is also interesting. And that is what we have. Now here, I wanted to show you this as well. Look at this. This is their phase 3 document. This document is, the link to this document is in the description of the previous video that says, will you take this vaccine? The link is there. So here, if you see, theoretical risk of enhanced disease, that is antibody dependent enhancement. Vaccine associated enhanced disease has been described for SARS-CoV-1 and MERS-CoV in some animal models and is associated with non-neutralizing antibodies. I have discussed that. I am going to discuss that phenomena individually again. Non-neutralizing antibodies and a T helper 2 secured immune response. So if the immune response they're saying is secured primarily on this side and less on this side, then maybe ADE may occur. In the animal models it occurred, not in humans. But still, if that is the case, then they're going to say, well, our vaccine actually is T helper 1 secured and our T helper 2 pathway is not really that big. So we are safe. In contrast, the AD26, that is their viruses, based vaccines have been shown to induce a clear T helper 1 skewed immune response and generate potent neutralizing antibody responses in both humans and animal models. So I thought this was interesting as well. So here we are. I hope it made sense, and I hope that you enjoyed it. So let's look at a couple of questions here and then we'll stop. We are already at 37 minutes. So there is a question from Brown Hilda. If it favors T helper 1 and as a result cytotoxic T cell pathway, is there a potential for cytotoxic storm? No. So the cytokine storm, there is no cytotoxic storm. Cytokine storm occurs because of the humoral pathway. So no. TD says, question, any way to enhance T helper 1 pathway? There are many ways. For example, if you see here, did I stop sharing? Let's see here. If you see here, this activation needs interleukin 12. Then this activation needs interleukin 2. And then the reverse activation needs interferon gamma. So if we theoretically, we give these interferons, then T helper 1 pathway will become active. And if we give interleukin 4, then T helper 2 pathway will become active. But normally, they don't suggest to modulate our immune system in this way because it can actually become a problem if the immune system arm incorrectly become stronger and weaker. Good question, though. So Shayur Ramdat says side effects. So we just talked about the side effects. The side effects are most common is fever, systemic, and most common local are pain at the site of injection, headache, fatigue, myalgia, and that's it. France says, wow, that is amazing. It seems to skew towards a good pathway. Correct. Correct. Shayur says, I am late to the discussion. How's this vaccine looking? Shayur, vaccine is looking good. And thank you very much. I just saw a super chat as well. So I'll go down in the comments and see who sent it, but thank you very much. Stop playing follow the leader. Kathy Miller says, stop playing, follow the leader. Okay. So Javier says, doctor, with this vaccine, somebody can have a cytokine storm? No. With any of the vaccines, we have not seen cytokine storms. But this would have even lesser chance of a cytokine storm because it is going cytotoxic pathway and not humoral pathway. Cytokine storm usually occur with the humoral pathways. Arun says, what is the role of adjuvants? Can we omit adjuvants as it is the main cause of anaphylaxis? How temperature causes inactivation of vaccine? Why vaccines are not given IV may be better. So let me answer all of those one by one. So can we omit adjuvants? In some vaccines, you cannot, because adjuvants' role itself is to kind of poke the immune system cells and trigger them to say, work, I am going to irritate you. In this vaccine, we do not have a lipid nanoparticle. We have an adenovirus that would itself act as an adjuvant. So I believe they don't have adjuvant. Moderna doesn't have adjuvant. They say our nanoparticle is sufficient. And of course, there is polyethylene glycol, which is causing an issue. That is the same problem with Pfizer as well. They do not have an adjuvant, but their lipid nanoparticle has polyethylene glycol, which can cause anaphylaxis. Can we remove it from there? I think no, because that is the structure of that helps make the structure. Then the second question, how temperature causes inactivation of vaccine? The higher temperature causes the RNases, the enzyme present everywhere that degrade RNA. They would break down the RNA. So they make it so much frozen that those little tracking machines of RNases do not function and they do not kill the vaccine or degrade the vaccine or decay the vaccine. That is why they keep it very cold. In this case, of course, we are not afraid of that. We just need slightly cooler temperature so that things work very sluggishly. But we do not have RNases. There is no lipid particle. There is no messenger RNA. So these are rather protected vaccines. Why vaccines are not given intravenous? We want the vaccine to be picked up by the tissue cells. And in the tissue cells are dendritic cells and the macrophages. If we give it intravenous, it will have to go somewhere, come out of the blood system, go into the tissue to then cause the macrophages to become active. Now, maybe it is easier if we give the vaccine in the intravenous system that it can actually leak out of the blood vessel. The problem is, imagine it's going to spread throughout the body and leak out of the blood vessel and all around the blood vessel. It is possible that the immune reaction starts, and that would cause serious vascular problems. So it is actually better to give it in the muscles. Abdul Razak says, good morning. Good morning back to you as well. Okay, so hopefully these are the majority of the questions. Let me just see a couple of more questions and then we stop. Brownhilda says, how can you say the T helper 2 pathway if you have a cytokine storm when it has IL-10 and the anti-inflammatory pathway? And T helper 1 causes production of interferon gamma? Very good question. So this is correct, that T helper 2, this pathway here, the interferon gamma pathway, amplifies the innate arm. The problem is that with the antibody side, the trigger that would occur when the antibodies start connecting with the cells and the complement system becomes activated, those triggers and the interleukins that are released as a result of that are much more dominant. Now, think about it for a second. Antibodies are released throughout the tissue. They go in the blood, they go in the tissue, and they would poke and trigger everything. On the other hand, cytotoxic T cell would just stay where they are. So they would not actually become billions of them and just run around everywhere. Well, they do, but not in the same quantities like antibodies. So this pathway is still much mellow in general. You are correct that interleukin 10 is given by the T helper 2 pathway. The problem is T helper 2 pathway interleukin 10 is not a big deal. More important deal is T helper 17 cells, which I'm not showing here, So whenever any pathway is activated, T helper 17 cells are activated, whose job is to suppress the system. Now, these T helper 17 cells do not function correctly in this behavior with SARS-CoV-2. And somehow that ends up in a cytokine storm. This pathway is not known to cause cytokine storm for SARS-CoV-2. So good question. So Shire says Novavax. So I've done a discussion about the Novavax. I would see if there is any further update. Lida says, how do the doses that J&J are studying here, 50 billion and 100 billion viral particles, compare to the doses in the other leading Western vaccines? Thank you. I do not know. I will have to do the comparison because not all of them are talking in terms of virus particles. For example, Moderna has messenger RNA and they do not have virus. Pfizer has RNA, they do not have virus. AstraZeneca has virus, so we can probably compare with that. Or CanSino has virus, we can compare with that. Shahida has a question. Was immune status of the cases and controls checked before giving the first dose? Yes. So they were checked for T helper 1, T helper 2. They were checked for reactivity to the spike proteins. So they were checked if they were already immune to them or not. And only those people were taken who were not. And so in their document, they actually talk about it, that we had tested for example, if you see here, they talk about that we had tested for T helper 1 and 2 cells beforehand. And probably here, I believe in this paragraph, they have talked about that they had done the T cell specific testing, and then they gave the vaccine, and then T cells increased. So it is here. I know I read it. Good question. This is like my test from the document to say, did you read the document clearly? Javier, thank you very much for the super chat. There was another super chat. I could not find the here. Sanjeev Gupta, thank you very much for the super chat. Cool. Bob has a good question on what day was the second injection given to cohort 1. Additionally, the 90% NAB figure you noted was for both cohort at day 28. Yes. However, the cohort 2 had slightly lower CTL response. Now the question, what was their dosage gap? I should have known this, but I have not seen it. So if I go to their method, I'm going to waste your time now. I'll look it up and I'll discuss maybe tomorrow. Procedure here, randomization, participant investigation to meet the criteria of building, sponsors, analysis, lab analysis, participants, vaccine safety, and this 403 grade 1 or 2. Let's see. Most we can, this one, the 2 cohorts. 1 or 2. I think they actually have not mentioned the day. That is why I don't recall it. Okay, I'll check it up again. I don't recall where they mentioned it. Tinkle Think says, can anybody answer? So I do not know what is the question. Simple Garden, they are saying the first early 2021. So don't know if early means Q1. I think it would be Q2 because they have to finish the phase 3 which just started, and then they have to accumulate the data, aggregate it, make sense of it, then go out for approvals. So my thinking is Q2. Denise says it is like a book report, but only for a study. Yes. And I'm not doing a great job. I forgot, or I have not read the gap between the 2 doses. That is a very basic question I should know. William, you're correct. Moderna has not. Moderna is, they are something. So, Faisal, that is a good question. Why does it take 30 days for the body to respond? Maybe because the dose is less. Remember, this is not an actual infection. In the actual infection, the virus just keeps going from one cell to the next and keeps increasing. Remember, in one cell, there are 10 million ribosomes. And imagine if they are making 10 million viruses, it is going to go from one to the next cell and then make 10 million more. And within thousands of cells, it's going to make billions and billions of copies, and we'll keep making it till our immune system stops it. That is not the case with the vaccine. Vaccine gave you 1 billion and done. So it probably, amount of dose is so little that our immune system has to kind of go slowly and work with whatever they're given. Okay, so later, even this one, they had the low and high dose, but then they decided for the low dose. Ivermectin, so you're talking about the ivermectin. We've been talking about ivermectin since March, April. It is, I think I was one of the first people who started discussing ivermectin after the study came out of Australia, and then Bangladeshi Dr. Alam's experiment or his usage of it. So yes, ivermectin is good. Cool. So this is the discussion for today. I'm just trying to see if there are. All right, so let's stop here today. Brownhilda, I'm seeing your question. Your question is, why is this explanation about this vaccine and explanation you have with vitamin D different? I thought the T helper 2 pathway was anti-inflammatory pathway, and the T helper 1, that is correct. So in the case of vitamin D, what we talked about was that vitamin D helps suppress the cytokine system generally. And so that is a good thing. Here, what we are talking about is that compared to T helper 2, T helper 2 is a humoral pathway. T helper 1 is a better pathway in terms of the cytokine storm not occurring. It is fine, it is pro-inflammatory, we know that. But we also know that when cytotoxic T pathway becomes active for SARS-CoV-2, even when it is pro-inflammatory, it does not lead to cytokine storm. Cytokine storms, I've done those studies in the past. Cytokine storm has been seen to occur in those folks who go T helper 2 pathway or humoral pathway. Why exactly they're trying to figure out what genes and what cells go mad? Is it the macrophage that becomes mad? They do not know clearly. But that anti-inflammatory pathway of antibodies actually leads to the cytokine storm in some people. And T helper 1 pathway, the lower pathway cytotoxic, is supposed to be anti-inflammatory. You are correct. Pro-inflammatory. You are correct in that. Look up Medicago vaccine. Okay, I'll check that out. So my cough has become much better, but I still sometimes get this reflexive cough. I have the HEPA filter going here. I have my humidifier going there. So the room gets cleaned more often. So cats come here a little less. So a little better, but still, whenever Luffy sits with me for a longer time, I start coughing. Cool. So let's stop today. There are some more questions here as well. I would look at them tomorrow. Let's stop for today. Tomorrow, if you like, we can talk about antibody-dependent enhancement. Thank you very much for your time today. Please like, subscribe, and share. If you wanted to buy me coffees, there is a link in the description to buy me a coffee. And if you just wanted to support my work as well, there is a link in description for that too. So thank you very much. I'll see you tomorrow.